Hello. Today I'm going to be discussing single-cell RNA sequencing, specifically Drop-seq, which was developed by Dr. Ivan Moscow and other researchers at Harvard University. The researchers published their findings in the journal Cell in 2015. The paper was named Highly Parallel Genome-Wide Expression Profiling of Individual Cells Using Nanoliter Droplets. Studying complex biological systems containing many tissues and cells has always been a question in gene regulation, and previous technologies were either very inefficient time and cost-wise, or single-cell analysis of transcriptomes was completely impossible. Studying gene expression at the single-cell level is key to understanding complex multi-tissue and cell systems. Drop-seq is a high-throughput method allowing for analysis and creation of many transcriptomes and can be carried out on a large scale efficiently. Results can be obtained in a day, and it costs only around 6 cents per cell. Drop sequencing is a method to associate specific genes associated with functionally specialized cells and how these genes and their protein products function to differentiate cell types and maintain cell specificity. Drop-seq will allow for large-scale analysis of mRNA expression in thousands of cells at a time. So in order to actually be able to quantify how many cell types are there, or how many unique transcriptionally unique things transcripts from cell types are there, they use this microfluidic chip system, where basically you have three lanes, and one of them is going to have cells from suspension coming in, and the other one is going to have microparticle beads with unique cell identifiers on them. And what's going to happen is that you're going to have an oil solution in the third inlet coming in and pushing it through and into the droplets randomly. And if the beads, so the microparticle beads and the cells are together in the droplet, the cells are able to lyse and RNA hybridization happens onto the microparticles, which were also in the droplet. Now, once this RNA hybridization has happened, we can turn these droplets into these things called stamps, okay? And these stamps stand for single-cell transcriptomes attached to microparticles, which I'll explain later. But these can be sequenced, and the cell origin can be determined using bioinformatic and statistical methods. So the researchers had to be able to have unique barcodes for each cell type, and the way they did this is to basically synthesize an oligonucleotide directly on the microparticle bead, which was then put into the droplet. And the functionality of this is to also capture the mRNA, for allowing for mRNA hybridization and reverse transcriptase priming. So the barcoded primer bead consisted of a UMI, or unique molecular identifier. The poly-T tail was for the RNA hybridization. All the beads have a PCR handle, which is for reverse transcription, and each bead also has the unique cell barcode, which is 12 base pairs, and all primers on the same bead have the same cell barcode. And the way this was done is random beads were each split into 4 groups with each having A, G, C, or T unique bases, as seen in the figure, pooled together and then split up again with the same bases, and this was done 12 times, and then the UMI was synthesized. So what really allows Drop-seq to work is this microfluidic chip technology. And the reason this is very cool is because it allows for these nanoliter droplets, which allows for a lot of scalability. Thousands of cells can be simultaneously done in one test tube, and 10 to 20,000 cells can be done per Drop-seq run. So just to give a small breakdown, in the figure provided, what I'm trying to show is that there's 3 inlets, which I had also talked about earlier, and one of them has a lysis buffer with the microparticles, and then there's a second one with this oil suspension, and then a solution of oil goes across that, and it kind of forces the droplets across randomly. But the problem with this is that only a few droplets actually have a primer and bead in a single droplet together. So for the droplets which do have a bead with the microparticle bead and a cell in the droplet, the cell is then lysed and reverse transcription occurs, and that leads to the creation of these stamps, which I had talked about earlier as well. These stamps can then be amplified using PCR. The stamps are used as a template, and then what happens is that there can be sequence alignment using paired end reads, and they use high capacity parallel sequencing. And those oligo DT primers on the barcoded beads is what allows for the reverse transcriptase to work. And they also tested the stamp organism specificity, which was not originally good, and they improved the lysis buffer and bead size so that the stamps would be highly organism specific. So once they had sequenced, and once the whole thing has been sequenced, all the stamps have been sequenced and aligned, unique cells and origin of genes can be found by creating a gene expression space, which I had shown a figure for on the last slide. This is very cool because now what they can do is actually visualize this gene expression space. But specifically in this study, what they were looking at are mouse retinal cells. And the stamps work because they're organism specific. And they use mouse retinal cells because they were already well studied. But molecular identifiers for some of the new cells haven't been found. So they were able to create 49,000 stamps in over 4 days and 7 replicates, which is really, really fast. And they were able to, first of all, not only match previous molecular biology studies showing retinal cell types in mice, but they also found new transcriptionally distinct cell types. To visualize this gene expression space that was created by the sequencing and alignment, what's used is a t-distributed scholastic neighbor embedding. And this allows for dissimilar data to be pushed away, which you can kind of see in the panel B of the figure, which is here. And you can see distinct cell types. And then this can be mapped, as you can see in panel A, kind of to a whole structure of a retinal cell. And this can be done for many systems. They just chose to use retinal cells for this study. So Drop-seq is a really powerful technology, but of course it does have limitations. The biggest being that only mRNA can be analyzed, and therefore only transcriptomic data can be really made from this technique. Obviously, one thing that could be done is to create a technology which still does single-cell RNA sequencing, but also can do it directly from the DNA. Competing technologies such as the single-cell RNA sequencing technologies developed by 10x Genomics have a higher molecular sensitivity. InDrop has a similar method, but these two are much, much slower and tend to be less cost sensitive than Drop-seq. Now, one way to improve Drop-seq would be to decrease the bead loss, which is currently really high due to the random nature of droplet formation, which results in a lot of empty beads. Additionally, since it is a very physical and manual process, things like pipetting can lead to droplets merging together, which means you can't even use them. And also constructing better beads, which are more organism specific, may be of use to research. Now, that being said, Drop-seq is a powerful technology and is currently being used to study a multitude of biological systems, and finding new cell types and new forms of regulation through cell specificity is really key. And understanding complex systems in varying biological systems depends on understanding cell functionality and specificity. And that can only be done using these very advanced technologies such as Drop-seq. And hopefully in the future, this can be also done directly from the DNA, and this will lead to a much higher understanding of genetic regulation and cell specificity amongst all biological systems.